in this section we will discuss about the experimental setting of the HPLC and UHPLC, ultra high performance liquid chromatography. So if you see the high performance liquid chromatography machine, it has mainly four parts. One is auto sampler. Second part is pump. It is otherwise called as quaternary pump. Quaternary pump. And third is column. Fourth is detector. So detector means we can divide it into UV DAD, UV diode, detector, FLD, fluorescent detector. So this is the main four components of the HPLC. So auto sampler is the area where the sample is taken. हम लोड किया ना ऑटो वायल में लोड किया वो सैंपल को 10 माइक्रोलीटर या 20 माइक्रोलीटर हम लेंगे. सो ऑटो सैम्पलर का सेटिंग के अंदर हमें क्या क्या करना है. फर्स्ट वी हैव टू डू इज इंजेक्शन वॉल्यूम कितना है सेट करना है. थोड़ा मेथड में हम 10 माइक्रोलीटर टू 20 माइक्रोलीटर तक इंजेक्शन वॉल्यूम ले सकता है. मैक्सिमम 30 माइक्रोलीटर. UHPLC मशीन अल्ट्रा हाई परफॉर्मेंस मशीन है तो मैक्सिमम 30 माइक्रोलीटर हम इंजेक्शन वॉल्यूम ले सकता है. सो अपने आप उस नीडल से वायल में जाके वायल से 10 माइक्रोलीटर या 20 माइक्रोलीटर निकाल के साइड नीडल वॉश इनेबल है हमारा. उसको नीडल को साइड वॉल को वॉश करके उसके बाद दूसरे जगह जाके अंदर इंजेक्ट करेंगे 10 और 20 माइक्रोलीटर. वो आई विल शो यू इन द मशीन. Then the next part is a quaternary pump. Quaternary pump means it will take 4 different A, B, C, D bottles over. Either inside the top of the HPLC there will be a bottle A, there will be a bottle B, there will be a bottle C, and there will be a bottle D. Here some bottle is connected to the quaternary pump via tubings. So we always use A is filled with water, B always be filled with buffer, and C will fill with the methanol NaOH, and D will be filled with the acetonitrile ACA. So it will be 100% always. It will be 100%. This also 100%, but this is adjusted to power, pH adjusted. This is always pH adjusted. So what will happen is once the machine configured, we have to set how much percentage of A is taken, how much percentage of B is taken, how much percentage of C is taken, how much percentage of D is taken. Usually in this close-up like method, we will take methanol 70% है तो water automatically default 30% आ जाएगा इसमें. So is a method selected, but in case of close-up, according to this method, I am taking a reference method. According to this method, we can select on reference example मैंने बता रही है. We can select acetonitrile 40%. So we will take from D. So A, B, C, D होगा. ये select कर सकते हैं अपने. So A में A is water ना. A में so I will use A rather than B. B I will keep it blank. A में हम use करेंगे almost 50% लेंगे. That is of 0.5% triethylamine and pH adjusted to 5 pH. pH adjusted to 5. This solution we will take A में. And B we will keep it as blank, and C we will keep it as इसमें इस मेथड के मुताबिक C में 10% methanol we will take. Okay, 10% methanol we will take. 10% 100 by 100 volume. Okay, weight by volume by volume. 100% purity, but 10% total percentage of the इसमें से 10% लेंगे, इसमें से 50% लेंगे, एंड इसमें से 40% ACN लेंगे. So that is also 100 by 100. It's clear now. So A is frequency में flow करेंगे. इसमें हमें क्या करना है, इतना सेट करना है. This is solvent gradient. This is isocratic flow, gradient flow, solvent gradient है. So मैंने solvent gradient सेट किया. उसके बाद इसके अंदर run time है. So run time हमें select करेंगे. I will select it into 10 minutes. Okay, run time मैंने सेट किया 10 minutes की. उसके बाद इसका flow rate भी है. Flow rate I will set into 1.2 ml per minute. 1.2 ml per minute मैंने सेट किया. So these are the three configurations we have to set inside the quaternary pump. उसके बाद we have to select the column. So we have already put the column as C18 and Hplc XDB column. Okay, this is the column of 150 millimeter length and 50 millimeter diameter, I think 50 or something millimeter diameter, 5 micrometer particle size. कॉलम मैंने सेलेक्ट किया. उसके बाद इस कॉलम के पैरामीटर एडजस्ट करने वाला 2 पैरामीटर. वन पैरामीटर इज टेम्परेचर, मेनली टेम्परेचर ओनली. वी हैव टू एडजस्ट द टेम्परेचर ऑफ 2 ends of column. Both ends of column combined करके we will select temperature into 35 degrees Celsius. Both ends of the column we will set the temperature as 35 degrees Celsius. So this is another experimental condition. And finally we will select for close-up some other compound we will use it. For pharmacy we can use UVD. But in FND के लिए we will use if a close-up we will use FLD, and FLD का wavelength we will set always. It is we will set it 230 or 232 nanometer wavelength as test wavelength and the reference wavelength as 340 nanometer as reference wavelength we will set. So this is the wavelength reference we have to set. So what are the parameters we set? Auto sampler injection molding set किया, quaternary pump में gradient set किया, run time set किया, flow rate set किया, then column select किया, column के अंदर temperature set किया, and detector select किया, detector के अंदर wavelength select किया. So these are the experimental conditions we need to configure and save this as a method in a file and folder. And then this method select करके we will run using single run command. In OpenLab software में we will go to single run control panel में जाके we'll select a single run select करके उसमें configuration A method select करके कितना time repeat करना select करके we will put the run. Then the run will be occurring first run. If we put 3 runs, first run, second run, third run, repeat 1, repeat 2, repeat 3 occurring, and we will get the chromatic runs. So this is a method by which we are configuring the experimental condition of the Hplc. And always auto sampler का temperature will be 20 degree Fahrenheit, 20 degrees Celsius is a default one, and pressure also a default one. Pressure is also adjusted, अपने आप adjust करेंगे, and it is a default one. High pressure only, but it is machine will configure अपने आप, so it is auto adjusted. So these are the things to be considered while running the chromator, means running the sample in the HPLC. Is it clear? Okay. You can, thank you. You can